Like I said, we've got two graduate student speakers tonight, Ryan Franke and Mikayla Haynes. Ryan is going to go first, and he is speaking about a fungal biological control for invasive black locust in Minnesota. Ryan grew up in Eagan, Minnesota, and learned to revere the natural world at a young age. During a fall camping trip around Lake Superior in 2015, he became fascinated with fungi and shortly thereafter joined the Minnesota Mycological Society. His fascination mushroomed after joining the society. He opened a gourmet mushroom cultivation business in 2017. And you guys may not know it, he was the editor of our newsletter from 2018 to 2020. So most recently, he's joining the mycology-focused forest pathology lab at the U of M in 2023. And he has presented to us on some other biological controls. So maybe we can get him to give us some updates on that. He talked about buckthorn earlier, and I'm going to put a link to his studies in the chat here. And welcome, Ryan. We're excited to hear about your research.
Thanks to everyone for having me back again. As a graduate student, every last penny counts for something. So I really appreciate the continued support of the club as I've been on this adventure of getting the PhD in my mid-30s. So yeah, thanks. A lot of gratitude for you guys. And tonight, instead of talking about buckthorn, I'm going to talk about a new project that we just got funding for. It started this past January to develop a mycoherbicide for black locust. So a similar aim as the buckthorn project, and similar methods are being used, but targeting a different tree. And so, yeah, without further ado, let's get into the meat and potatoes here. So first off, I just want to talk a little bit about black locust in case you're not familiar. It's also just a very interesting tree with interesting history and ecological traits. And so first off, basically it's in the legume Fabaceae family. So it's related to beans and peas. However, this is a large tree, and it has deeply furrowed bark. It has these kind of orange tone in the furrows, and it has pinnately compound leaves. It can grow typically to 40 to 60 feet at maturity. However, it can reach up to 100 feet in excellent conditions and with no disease. It has both a shallow and deep root system, and the architecture of the roots and its depth in the soil depends on the soil conditions. And so in arid environments, it can grow really deep tap roots, up to, gosh, I read like 20 feet down into the soil, which is pretty deep. But also it can form this really vast network of shallow fibrous roots too, to take advantage of that profile of the soil. And this tree can reproduce asexually via root suckering. And so you can see the photo on the left here shows a root, also called a rhizome, which is basically a lateral underground stem at the bottom of the picture here. And from that rhizome, you can see these shoots coming up. And these shoots will penetrate the soil line and grow up into the atmosphere and grow leaves. And so this is similar to an aspen, that kind of growth form. And so you can have these enormous individuals that you may see 100 or 200 stems, but it's all the same individual tree. And here's the one and only black locust tree on the UMN St. Paul campus. You can see it has a pretty nice form. It's a medium large size tree. And like I said, it has clonal propagation for spread, but also it can spread pretty aggressively with seeds. It produces annually a very large seed crop, and they're produced in these pods here. These are at the end of a growing season. And then the following spring, the seeds pods break open, and then the seed pods and the seeds are wind dispersed. Um, and there can be, tens of thousands of seeds on an individual mature tree. And these seeds can spread and germinate well on disturbed ground. And so this can be, where there's been some human development, like along roadways or in neighborhood developments, also in old fields, old agricultural fields that aren't being planted. Black locusts can move in. Also in degraded woods, where there's been perhaps some earthworm activity that reduces the leaf litter layer on top of the soil and exposes some ground, um, and these black locust seeds can germinate and take root and start growing. Some other interesting characteristics of this tree is that it has large thorns, and these are extremely pokey. And so whenever I work with this plant, I wear long sleeves and thick gloves. I've gotten poked a few times, and it really does hurt. um, and so, yeah, buckthorn has a thornier name, but this is definitely a more treacherous tree to work with. And not only that, but its stem, leaves, bark, and seeds all have toxic compounds, these toxyl albumins, both rhobanin and rhobatin. And if you ingest any parts of those tissues of the tree, you can experience basically gastrointestinal upset, but also nervous system disorder. And this is especially acute in horses and can be fatal for horses. And there's some literature out there describing that. However, very curious that the flowers are edible. And so I've eaten these myself, and they're delicious. You can't miss these flowers. They're a later flowering tree, but then again, trees flower early on in the season, and so these black locusts in Minnesota, at least in the Twin Cities, just finished flowering. I'd say maybe a week and a half ago, and so they usually flower in late May, and the flowers have a beautiful aroma. They are favored by pollinators like native bees. And also they have a sweet kind of pea-like taste. And so you can make a salad that the base is these flowers and it's delicious. However, just make sure you don't accidentally pluck any leaves as you're plucking the flowers, because the other parts of the tree are toxic. So you just have to make sure you wear your glasses when you're out harvesting these flowers. Now, also interesting, so this is in the Fabaceae family, and this tree does in fact fix nitrogen. So it has these little nodules that form on its rootlets in response to infection by a rhizobia bacteria. And inside of these nodules, there's basically a decreased concentration of oxygen. And in that more kind of less aerobic environment, the bacteria can efficiently transform atmospheric nitrogen into bioavailable nitrogen. And so these trees can and do enrich the soil with nitrogen in which they're growing. Also, the wood is extremely dense. It has a BTU value between white oak and Osage orange. So it's one of the best firewood that we have in the country. And the wood is also very rot resistant. The heartwood has a lot of extractives like phenolics and tannins in it that make it difficult and recalcitrant to decay. And so you can see people will do things with this wood with outdoor applications like making a mailbox post out of a round of black locust. Also, people can make basically finished wood products out of black locust. And this is a website, robodecking.com, that sells these 4x4 black locust posts. And I think they were like a 4-foot post for $20 or something like that. So fairly affordable, and it looks really nice, and you don't have to put any kind of chemicals on it to treat it. So it's a nice natural option. The most interesting thing about black locust, and perhaps also most surprising, um, is that it is fluorescent. It has fluorescent wood. And so you can see that the sapwood here on the outside of the stem here fluoresces this blue color, and the heartwood fluoresces this highlighter yellow. And you can see the pith in the middle doesn't fluoresce at all. So when you shine a black light over that wood, you get this beautiful fluorescent show. Um, and this is something when I first started working with black locust, my advisor Bob Blanchette said, hurry up and split me a section of of that wood, because he has I would say he's a fluorescent hobbyist. He likes finding fluorescent things in nature. And he hadn't yet seen black locust, and so we split this open in the lab and turned off the lights, and we were pleasantly surprised with the show of fluorescence. A little bit of the history of this tree in North America is pretty interesting, both from an ecological and a human history standpoint. So the original range is, you can see depicted in the map on the left, and it spanned kind of two distinct areas. Further in the west, we had black locust growing in southern Missouri and in Oklahoma and Arkansas, in the mountainous regions in those states. And then further east, we have a range in the mid-Atlantic area, stretching from Alabama and Georgia in the south up to Pennsylvania in the north, in the Appalachian Mountains. And this tree thrives in disturbed areas, and so it's kind of the equivalent of like our aspen here in Minnesota, where it's very fast growing. It's often first to a site in its native area, and then grows very fast, and it has a sparse canopy, and so other native trees and plants are able to survive in its shade, and then grow up underneath the black locust and replace the black locust in succession. And let's see what else to say. So from a human history standpoint, this tree was favored for timber production in the United States back in the 1800s and early 1900s. And so much so that in 1930s on Long Island in New York, there was a discovery of a clone of black locust that had no fork in the bowl. And so normally black locust does fork pretty low in the bowl, which makes it less suitable for a timber production. But this clone had no fork in the bowl, and so this was deemed the shipmass locust variety. And it was cultivated and propagated clonally and on the East Coast. However, by the mid 20th century, most of the black locust timber production in our state was being marketed and sold as fence posts, because the chemically treated pine had replaced its other outdoor applications for building, and so it just kind of became less planted. However, in the 1990s, some Polish scientists began breeding black locust because they figured that it actually grew really well in Eastern Europe, and they bred a variety that they named shipmast. And there's actually quite a few varieties of black locust that are widely cultivated in Eastern Europe today for timber. Back here in the United States, today this tree is still used for human applications, specifically to remediate old strip mining sites. And so basically these trees are able to grow on very poor mineral soil that doesn't support other trees, and they're able to stabilize these strip mine tailing piles, and then through fixing nitrogen, they enrich the soil and create habitat for other tree species to move in the process of succession. And so it's still a fairly useful tree even today. And let's see, move on to the next slide here. So the question that a lot of invasive plant specialists in Minnesota and Wisconsin and I guess in the Midwest are asking is, is black locust invasive, or perhaps is it native because it grows in the United States, and so does that mean it's native in Minnesota? And while you can see, clearly it's moved well beyond its historical distribution. It's even growing on the West Coast. And here in Minnesota, we are on the kind of advancing front of black locust as it sweeps across the country. And the issue with black locust is that because it can grow on dry, nutrient-poor soils, it's able to grow in habitat that historically has supported like oak savanna adapted plant species. So these are plant species that are not competitive in high nutrient environments. However, black locust moves in, outcompetes them because they grow over the top of them, and then they enrich the soil with nitrogen, and the allows other plant species to begin growing there that then outcompete those specialist prairie and oak savanna adapted species. And so it is a problem, and also it's really hard to remove black locust because it does propagate via rhizomes under the soil. It's really good at resprouting after it's been cut. And so you can see here, this is a tree that was cut, and it grew not just one, but four new shoots. And these aren't just little shoots. These are 8 feet tall, and so it responds really well to cutting. And so different herbicides are being developed to try and be able to manage this tree, and even chemical herbicides fall short. And so it's a thorny problem right now. However, it has a lot of useful traits, as I've been mentioning, and it's actually by area is the second most widely cultivated tree across the globe. A lot of places, it can thrive in a lot of places in temperate areas, and it has utility, and so different societies grow it. And in fact, 20% of forested land in Hungary is planted with black locust. However, also in Europe, it also has spread beyond cultivation and also poses a problem in habitats over there. And so it's it's kind of a tree with it has some beneficial traits, but also it can be problematic. And so why this tree is being grown for timber in Europe and is really one of the premier timber species in Eastern Europe. And so why not here in North America? And really the reason is because we have an insect pest, the locust borer, that is a specialist on black locust. And this beetle will feed on goldenrod exclusively in the late summer, and then it'll go and lay its eggs on black locust. And those eggs will hatch, and those larvae will tunnel into black locust and into the heartwood, and they'll eat the heartwood and the sapwood basically all fall. And then in the following spring, they'll eat some more, and then they'll pupate and they'll emerge in midsummer and go and feed on goldenrod. And you can see here's a fluorescence of black locust in the cut section, and you can see those locust borer holes in the middle of the cut section. And these holes and the entry points of those galleries serve as infection courts for fungi. And so the most prominent of which is the Phalaenus robineae. Let's see, do I have that next here? Yeah. And so this is a Phalaenus species that specializes on black locust, and it enters through the locust borer galleries, and it rots out the heartwood. of the tree. And so unfortunately, the locust borer will target trees that are as small as 4.5 centimeters diameter breast height. And so even young trees will basically get infected with this heart rot fungus, and the whole center column of the tree will be rotted out. However, the locust borer is not found in Europe. And so they're able to grow this tree without, the most pervasive problem that we're dealing with here in the United States. Additionally, we have a fungus here, Vanderbilia robiniophila, that specializes on black locust and also can infect hackberry. And this produces a butt rot of the tree. And this is the fungus on hackberry here. We also have lots of canker fungi here in the United States that cause branch cankers and limb and stem cankers on black locust, including Diaporthioncostoma and Nectaria cynobarina. And you can see here, I haven't yet isolated the fungus out of this canker, but here we have a target-shaped canker. So we have the center of the canker is the portion of the stem that was killed first. And then the tree formed this callus tissue around the diseased tissue, but the fungus was able to overcome the tree's defenses and kill that callus tissue. And so you have the next layer of the target-shaped canker, and you have more callus that was killed, and on and on. And so you get this neat-looking canker. And this is typically a signature of Nectaria datissima that has a pretty wide host range and can form cankers on oak and birch and sweet gum. And apparently it looks like black locust as well. Also, there's this fungus, not in Minnesota, but in black locust more southerly distribution in Arkansas and Oklahoma, this Texas root rot. And this is a problem down there for black locust. And so the reason why I'm here talking to you about black locust today all begins with this buckthorn field experiment that myself on the left and the undergraduate research assistant, Evan Worrell, on the right, and I were putting in Cottage Grove in 2024. And we're inoculating buckthorn trees. And we noticed over our lunch break that across the road from where we were, there was a black locust clone that had a lot of dead trees in the clone. And so we had to investigate. And, we had our handsaw and other tools. And so we could investigate and take samples and chop into the wood, which was just, a lot of fun to do. And we saw when we got into the stand that a lot of these trees had these huge elongate cankers, and they either threatened to girdle the stem, or they did girdle the stem and kill the entire tree. And you can see inside of these cankers in the dead wood that was exposed by the canker, we have these wood decay fungi growing out. Here we have this wrinkled peach, Rhodatus palmatus, fruiting body here at the top, and a little one coming out at the bottom. But we also saw some turkey tail and Trametes hirsuta and some other wood decay fungi in these cankers. And so in addition to this site, I was driving down the highway later that summer, and I noticed another black locust clone that had some dead individuals. And so I went and took samples at the second site. And with the samples between the two sites, I had maybe like 20 sampled trees. And from those trees, I ended up isolating about 50 different fungi. And I was looking at these fungi and was searching them in the scientific literature, and found that a few of them were implicated in causing serious disease in black locust timber plantations in Europe. And so, there's not much plant pathology work that's been done on black locust in the United States because it's not really of economic value here. But in Europe, there's been quite a bit of plant pathology research done on fungi causing disease in black locust stands. And some of the fungi that we found here in Minnesota have been shown to cause serious disease on black locust in Europe. And so I thought, hey, this tree is an issue here. Maybe we can use some of these native fungi to try and manage its growth in sensitive ecological areas. And so that was born the project, and we wrote a grant and got a grant to develop a microherbicide for black locust. And so let's see, how am I doing on time here? Pretty good. Okay. And so the project basically follows these 4 steps. First, we're going to collect fungi from diseased black locust, and we'll be doing that this summer. That's kind of our focus this summer. And then once we have a large collection of fungi, we'll be testing those fungi for their ability to cause disease when inoculated into healthy black locust in pathogenicity studies, both in the greenhouse and in the field. And then we'll be developing methods for applying these fungus to black locust in the field. And lastly, we'll monitor the effect of the microherbicide on the plant communities where the microherbicide is being used. So if you kill the black locust or affect the black locust with the fungus, how does that affect succession after the fact of the management? Okay, so getting to, we have the most data I have is for the first step, the collection of fungi we had from those first 2 sites that we sampled in 2024. And so let's look at that here. And so this is the abundance of the fungal species that we isolated from those 2 sites from black locust. And the bigger boxes indicate that we had more trees from which we isolated that fungus. And so the left-hand side of the figure here, we have a couple of fungi, Diporthioncostoma and Neocosmospora metavorans, from which we isolated from 5 different trees. And then the smaller boxes over on the right are fungi from which we just isolated from 1 tree each. And then we have some fungi in between. Now, Diporthioncostoma is an interesting fungus because it has been implicated in serious canker disease in black locust in Europe. And that was, came out of 5 different trees. I think it was 3 trees at the site in Woodbury and 2 trees at the site in Cottage Grove. And then also to note is this Camarasporidiella ribinicola, and this has also been implicated in serious canker disease in black locusts in Europe. Some other fungi here, we have Urpex lacteus. This is a canker rot fungus that both cankers and can rot wood simultaneously. It's pretty common. It's known as the milky white tooth polypore. Pretty common on like dead branches and on snags. And then we have the Phelinus robiniae. This is the heart rot of black locusts that we saw a few slides back. Now, we grew some black locusts from seed starting in 2025. And so those black locusts were about, 5 feet tall this past winter. And we conducted our first pathogenicity test on those trees. And we used 8 fungi from our buckthorn collection. And what we did was we cut these black locust trees to stumps, just about an inch above the soil. And then we grew our fungi on sterile paper towels. And once those paper towels were myceliated, we put those myceliated paper towels over the cut stumps and then wrapped it with parafilm to prevent the inoculum from desiccation. And then we recorded basically the number of stump sprouts and apparent mortality 12 weeks later. And the only fungus from this study that appeared to have an effect on the black locust was this Fusarium sambucinum. And so you can see our control inoculated stems. So these were stems that were just covered with a sterile paper towel are on the left, and our Fusarium sambucinum inoculated stems are on the right. And this was 4 weeks after we inoculated, cut and inoculated the stems. And one of the first things you learn in plant pathology as a student is that plants, have limited amounts of resources, and they can kind of balance their allocation of those resources according to their environmental conditions and stressors. And so they can either shunt those resources to growth and reproduction, or they can shunt the resources to defense. And there's always, some resources going in either direction, but depending on, if they're dealing with a serious pathogen, then the plant will grow less because it's spending more of its resources and energy and producing secondary metabolites to prevent the growth of the pathogen. And so you can see that the control inoculated stems have lots of growth after having been cut just 4 weeks after that. However, the fungus inoculated stems have essentially there is no shoots after just 4 weeks. And some of those stumps actually didn't sprout for 10 weeks. And one of them never sprouted. And so we isolated, we took a little tissue section from that dead stump and we put it into some agar media and we isolated Fusarium sambucinum out of that dead tissue. And so clearly it's capable of causing pretty serious disease and even mortality in stems. Additionally, 2 of the 10 stumps inoculated with this fungus have sprouts, but they're only 3 to 4 inches in height. However, in comparison to the control stumps have sprouts that are 2 to 3 feet in height. And so, this isn't I guess, a 1 in 10 mortality rate, a 10% mortality rate is not very impressive. However, working with buckthorn, I know that fungi that are very pathogenic on buckthorn in the field cause much less severe disease when inoculated into buckthorn in the greenhouse. And I think the reason could be that trees in the greenhouse have no shade, so they have 100% light availability. We give them fertilizer, slow release fertilizer, and they're watered every day. And so they have a lot of resource from which they can use to mitigate the effect of the pathogen. And so I think actually having one stump that was killed outright is actually pretty promising. And I should mention too that this fungus has been implicated in wilt in black locust stands in Eastern Europe. And so, yeah, we were excited to see some promising preliminary results here. And so we selected Fusarium sambucinum for a second greenhouse study. And so we had some more black locust trees, and we decided we were going to inoculate those trees with the propagules of Fusarium sambucinum. And so the Fusarium genus species often produce these asexual macroconidia, and they're usually canoe-shaped like this. And these are infective propagules, and they are produced in massive quantity when you grow the fungus on mung bean agar. And so we basically boil some mung beans and take the filtrate and add agar and sterilize it, and then pour agar plates with the mung bean nutrients agar, and it produces a lot of macroconidia. And so we were able to basically wash the macroconidia off those mung bean agar plates and put them into suspension, and then cut black locust to stumps, and then put some of that macroconidia suspension onto the stumps. And we did that this past spring. And so I have 2 undergraduate research assistants working with me on this project this summer. Vanessa Kelly here is helping me inoculate, and then the photo was taken by AJ Jensen, who I know is on this call and is very excited about this project. So shout out to AJ for all your help. Okay, and so a way that you all can get involved with this project is by helping us locate dying black locust in Minnesota and Wisconsin. And if you find dying black locusts out there, we have a Google form that you can use to report its location. And so this is a QR code that links to that form. And what we're prioritizing are trees that have wilt symptoms. And so we're really excited about the prospect of finding a wilt pathogen, because wilt pathogens move very fast in the tree's xylem and can kill a tree very fast. The entire tree, just like Dutch elm disease, kills the tree quickly, or oak wilt kills the tree quickly. And so, we're not unlike Dutch elm and oak wilt, we're not looking for an invasive pathogen. We're looking for a native wilt pathogen that can be used to control this invasive tree. And so here's, for reference, a photo. This is honey locust wilting, but something like this, where you have the foliage basically dying, drying out, or I should say, usually turning yellow first, and then drying out and brown very quickly over the course of maybe just a couple of weeks. And so this is what we're on the lookout for, this kind of symptomology. And if you see this, or if you see other symptomology too, go ahead and report that on the Google form. And yeah, this whole project is funded by the Minnesota Invasive Terrestrial Plants and Pest Center, which in turn receives funding from the Minnesota Environment and Natural Resources Trust Fund, which receives money from the LCCMR, which gets its money from the Minnesota State Lottery. And so, yeah, if you go out and buy a lottery ticket, you're helping support lots of projects like this, which is kind of a neat way to use our public funds. And a lot of people have been helping this project move forward. First and foremost, my advisor, Bob Blanchette, professor at the University of Minnesota. He's the principal investigator on the project. Also, Ben Held is our lab manager and makes sure that we have all of the lab supplies and field materials in order to do our research. Undergraduate researchers, AJ Jensen and Vanessa Kelly, are, they're in it with me. They're in the trenches, and every day we're either isolating fungi or preparing growth media or inoculating trees in the field. And it's not easy work, but it's also a lot of fun. And so it's nice to have good people to do this fun work with. And yeah, and then a whole bunch of people, Alex Roth, Sasha Lodge, Monica Chandler, Dan McSwain, Brad Gordon, they all provided letters of support for our grant proposal. So many thanks to them for helping us get across the finish line for our funding. And last but certainly not least, the Minnesota Mycological Society for supporting my work for the past 3 years since I've been in grad school. Yeah, many thanks. And yeah, with that, hopefully we have some time for some questions.
Thanks so much, Ryan. We can't wait to hear about the results too. I've got 2 questions in the chat right now, but folks can put more in as they think of them. Peter's asking, are there any mycorrhizal fungal partners with black locust?
Oh man, not that I'm aware of. There could be, but I think that because they're the Fabaceae family, I think they're maybe less likely to form mycorrhizal associations. Yeah, I don't know of any off the top of my head.
Interesting. And then Peter's also asking, is honey locust invasive?
I don't think so. I think it has a historically more northern distribution. Yeah, I don't think so. Also, they're in the same family, but they're not in the same genus. And so honey locust is Gladitzia, and black locust is Robinia. And I'm not sure if honey locust reproduces asexually via rhizomes. And that might be why it's not invasive. I'm not sure, but I know it's not a problem.
And then Heather had mentioned, I think a couple of years ago, we had a presentation on assisted forest migration. And just because of climate change, things that usually grow in certain areas are moving to other areas. Or actually that presentation was about, helping plants move to those areas knowing that it's going to be suitable. But do you think the climate has anything to do with black locust spreading, or do you think it was inevitable?
Yeah, I mean, I do. I think the climate does have to do with it spreading. That, the ranges of the trees in North America have kind of changed over millennia. Over geological time, the ranges look very different with the warming and the cooling of the climate, because the climate's always warming. It's going through warming and then it's going for cooling, going from warming to cooling. And so yeah, I think that would it come up? Yeah, I mean, I think it is probably a natural process. Was it also potentially assisted by people finding usefulness in the tree and moving it west? Yes, I do think so. It was often used for like windbreaks because it grows really fast. It can also enrich the soil um, and so I think it's kind of both, that it's been human assisted in the past, but also the warming climate is favoring its northward growth too.
Interesting. Very interesting. Yeah, our neighbor had a huge one in her yard, and it was kind of, always dropping stuff onto my garden, which was already tilled and ready for it to plant. And I always kind of, at first I felt lucky because I was like, oh, the flowers are so pretty. And then after I had to cut a bunch of them out and noticed how big the thorns are, I wasn't feeling so lucky anymore.
Yep. Yeah, you got to wear thick gloves when you work with the plant.
Does anyone else have any other questions for Ryan? Please feel free to unmute as well if it's easier. Oh, I just thought of one thing, and I was just thinking about how you showed the wrinkled peach on that one. And it seems like a lot of those other mushrooms that grow that are the wood decayers, they're just waiting for something to be at the right stage of decay so they can take advantage of it. Is that correct? So it's not specifically that it was a black locust. It's just that the canker had attacked it. And so it was decayed enough that then the wood decay fungi could kind of take their turn. Is that correct?
Yes. Yep. Yep. And so the sapwood becomes available because it's kind of like dead, and so then the wood decay fungi can get in there. They have a harder time with decaying the heartwood because the heartwood has so many extractives in it, and only select fungi are adapted to degrade that, like the heart rot fungi. But the Chermites versicolor and hirsuta and wrinkled peach can get in there and decay the sapwood. So yeah.
Cool. Very cool. Awesome. Well, thank you again. Yeah. Very exciting.
Yeah, absolutely. Thanks for having me.
I want to take this time to introduce our second speaker tonight. It is Mikayla Haynes, and she'll be presenting mycorrhizal fungi recruitment to seedlings, tree to tree, or wind dispersal. And Mikayla is a third-year PhD student in Dr. Julie Ederson's lab at the University of Minnesota, and has spent her time in the Integrated Biosciences program researching ways that mycorrhizal fungi can benefit reforestation efforts. As a lifelong Duluthian and fungi enthusiast, she has enjoyed getting to know her fungal neighbors, both edible and the cool ones, observing responses of known fungal species to different precipitation patterns throughout the years. And in learning about the importance of mycorrhizal fungi, she's dedicated her current path to identifying habitat restoration opportunities in northern Minnesota, where she can implement mycorrhizal fungi application to improve and restore landscapes to functioning habitat for sensitive plants and animal species. And I'm going to put a link to Mikayla's work in the chat. And welcome, and thank you for being here, Mikayla.
Yeah, thank you for that lovely introduction. As stated, I'm Mikayla Haynes, third year PhD student, and today I'm going to be talking about mycorrhizal fungi recruitment to seedlings, tree to tree versus alternative dispersal. I decided on alternative rather than wind, just because we don't necessarily know whether or not it's truly wind dispersal or if there's other things helping out. So a little bit more about me. There's all the things as stated before, but largely in my PhD studies, I've been doing greenhouse-based experiments. And now one of the experiments that I'll be talking about today that the Minnesota Mycological Society is funding the DNA sequencing for is my farm-based experiment. After my undergraduate degree, I worked in ecology and habitat restoration for 6 years. And then I went back to school because I wanted to kind of pursue my own path and be able to um, basically write grants for projects that had to do with fungi. And that was through Lake Superior Research Institute out of UW Superior, which is across the bridge from Duluth. And yeah, I'm a lifelong Duluthian. I've never really wanted to move away, and I kind of want to keep staying here. Now I have a bunch of friends moving back, so I feel like I made the right choice. Yeah, and then that's the biggest chicken of the woods I've found, so that was a fun share. All right, so we're going to get into mycorrhizal fungi. I know many people here know so much about fungi, but I'm just going to go through the basics of mycorrhizal fungi to cover my bases. So we have myco and rhizal, which myco is fungi, rhizal meaning root, because they associate with roots. The volume that a tree or plant can reach on its own is known as the rhizosphere. And then with its fungal partner, we can see that that increases the soil volume to up to double the soil volume that a tree can reach on its own. And this is known as the mycorrhizosphere. This symbiotic partnership aids in nutrient uptake. So the tree provides carbon to the fungi via sugars that it creates with photosynthesis. And then in exchange, the fungi breaks down more difficult to access nutrients like phosphorus and nitrogen in the soil and gets that to the tree. And they're happy together that way. It also helps with drought resilience. The mycorrhizal fungi has smaller mycelium than the root hairs of a plant can reach in the soil. Therefore, it can access smaller soil pores. And also, with the larger volume of soil that it can reach, it might be able to access those water reserves that are tucked away into the soil. It also helps with soil stabilization. Mycorrhizal fungi excretes enzymes that bind soil particles together, which can really help with soil erosion or reestablishing nice soil aggregates after a disturbance. And then it also helps with pathogen defense. So whether that's microbial pathogens or other fungal pathogens in the soil. There are different mycorrhizal fungi types. So typically what we'll see in like a prairie situation or some trees, largely fruit trees, are buscular mycorrhizal fungi, also known as endomycorrhizal fungi. And this is because the fungi hyphae goes into the cells themselves. You can see here it's entering into the cell, and then it forms an arbuscule, which is just kind of a cluster of hyphae within the cell. And so it's kind of a direct pipeline from the outer cell to, or from the soil environment into the cell. There's ectomycorrhiza, which typically associates with trees, especially trees up the North Shore here, as we see. So we have a lot of pine species, conifers, and it also associates with species like yellow birch and paper birch and northern red oak, etc. There's ericoid mycorrhiza, which associates with the Ericaceae plants, which includes blueberries and bilberries. And those also form hyphal coils within the cells. There's orchid mycorrhiza, which associates with orchids, as the name suggests. So something like yellow lady slippers or any of the lady slippers might have that up north here. And again, that goes into the cell. And then we have our buttoid mycorrhiza, which associates with largely understory plants within the forest. For me, I focus on ectomycorrhiza, and this is partially because it's a lot easier to work with under a microscope and identification-wise. There's no staining. I can just identify them by sight. And they're also easier to harvest because they produce these lovely things known as mushrooms. So what about commercial products? If you've been paying any attention, you've probably started to notice that mycorrhizal products have entered the I don't know, the product line of gardening and different forestry practices. This one, Myco Apply in particular, has the most native species that I've encountered, but many of them aren't necessarily native species. They're just kind of whatever's easiest to grow. Many are endo or arbuscular mycorrhizal fungi, not necessarily encompassing that ectomycorrhizal fungi aspect. They typically have a relatively short shelf life where they do expire. So you can buy a bunch and not necessarily use it all, and it's quite expensive, but then it'll expire. And what are you supposed to do with it now? It may not contain species that they claim to contain. There are several studies that have done testing on these products, multiple products, and hardly any of them had 100% of the species that they claimed to contain. The fungal species may not be compatible with the plant species that you're targeting. So just because they are widely compatible with plant species doesn't necessarily mean that they're compatible with the species that you're targeting. And this is where the high cost on the market, the marketing, and then people thinking like, oh, this is great. I'm going to buy this product and then inoculate my plants. This is where this can kind of have a mismatch. And furthermore, non-native fungal species could have negative impacts. So if they're not native, they're not compatible with the species that you're targeting, what are they compatible with? There are multiple papers that explore the invasive impacts of mycorrhizal fungi products, and there's evidence of co-invasion effects where a plant might not necessarily be native, but it doesn't have its mycorrhizal fungi partner, and then it encounters its mycorrhizal fungi partner, and all of a sudden it has all of it needs to succeed widely on the landscape, and therefore goes forth and does that. Going into my experiments, I have several different experiments that I've conducted. I'm just going to kind of go through these so you know what the base is and how I kind of came to study what I'm studying right now. So my first experiment, which is complete now, studied commercial, native, and control. So I had Myco Apply, which was that mycorrhizal fungi. Again, I selected for it because it had the most native species out of all of the mycorrhizal fungi products that I had available to me at the time. Of course, new ones are coming out every year, it seems. I took native mycorrhizal fungi via a soil slurry. So I collected soil from the sites that the seeds were from. I had control, which I autoclave sterilized potting soil, and then didn't inoculate anything. I had 5 tree species. So I had pin oak, sugar maple, northern red oak, eastern white cedar, and yellow birch. 1, 2, 3, 4. I think that's 5. And then, yeah, I only measured the above ground traits, which I later on, you'll see that I did do below ground traits. But this was to inform a farm-based growing project. So if anybody's heard of the Minnesota Million, we partner with farmers. There's a farmers cooperative where they're taking on the task of growing tree seedlings to try and supply more trees to the Minnesota Million project, which is trying to plant more trees up to 1 million acres in Minnesota. So they need quite a few more trees than can be provided by the state nursery. What I found with this one is that, or with this project in particular, is that really only yellow birch had significant results where the commercial mycorrhizal fungi actually outperformed native or control. And then we frequently saw that native mycorrhizal fungi performed more poorly than even the controls did. And again, we didn't do below ground traits. We didn't do any DNA analysis on this. So it kind of seemed like, well, maybe there's something else going on, like perhaps there's soil pathogens. Either way, it was a stepping stone to learn more and apply those lessons going forward. Timing of inoculation was a kind of sub-experiment. It was an undergraduate student of mine. He conducted this experiment with my guidance, Jacob Pierskala. This was conducted on eastern white pine, and we took native soil from a reference forest that was nearby, this forest in particular. It's Magny, I believe, if anybody's familiar with that forest in Duluth. And we inoculated white pine seedlings grown from seed at different times. So it was 4 weeks into growth, 8 weeks into growth, 12 weeks into growth, and then not at all. And this informed my cross inoculation experiment later on. This is a photo of the mycelium extending from a colonized root tip, whereas this is an uncolonized root tip. So you can see how that kind of affects the growth and then how obvious that is under a microscope. But what we found is that, kind of as expected, if trees are inoculated earlier, they have a higher root colonization percentage versus later. But something that we didn't expect is that there's an inverse relationship between the colonization and the root to shoot ratio. So those inoculated later or not at all had much larger root systems than the trees inoculated earlier. And this is quite interesting because if people were to apply this, if nurseries were to apply this, what would you prefer, a well inoculated tree with a smaller root system or a large robust root system? Even though there's no difference in above ground growth, these are just things that should be considered. Either way, this informed my next experiment as to when we should inoculate it. So everything's inoculated at the same time, with ideally the most colonization to see if there's any growth effects, which was the big cross inoculation experiment. This is mostly complete. My eastern white pine portion of the project is complete. I had eastern white pine and yellow birch. They're two prominent species along the North Shore. I conducted my project within the Minnesota Lake Superior Coastal Zone, and I'll get into this a little bit more later. But basically, I took 5 seed sites, 5 soil sites, and then crossed them all. I was evaluating the local adaptations of mycorrhizae with soil slurries. So does it matter where the soil comes from? Do the trees value what their mother experienced as far as mycorrhizal fungi community goes? I looked at both above and below ground traits. So I did root colonization and root to shoot ratio for these, as well as height, stem diameter, and crown, I believe crown diameter. For the yellow birch, I found quite a bit of colonization within the controls. So I'm doing DNA sequencing to see what the background communities are of the greenhouse. greenhouses are not sterile environments, and quite often in both the pines and the yellow birches, I would see cup fungi and conidial mats forming within the cones, which turns out they're mycorrhizal. So got to figure out what species are kind of dominating those controls to figure out what actual communities and those effects might be. And then that brings me to the Tree to Tree project that the Minnesota Mycological Society is helping fund. That's in progress. I have both nurse and bait trees, which I'll get into a little bit later. This is farm-based, so this is just a tiny plot out at the UMD farm up in Duluth. And I am looking at above ground traits, and then for below ground, I will be doing root to shoot ratio as well as quantitative DNA. Microscopy is fantastic, and again, you can kind of see here, these are a different type of colonization, but they are indeed colonized. But it's extremely time consuming to do microscopy, and at the end of the day, we with the amount of hours spent with microscopy versus DNA cost, we figured out that DNA is actually cheaper than microscopy for large sample sizes. So again, I have the cross inoculation that I'll be talking about today, and then the tree to tree. So as far as local adaptations, can anybody tell me the biggest difference they see between these two photos? I've been a TA for a couple semesters, so asking questions is normal for me. But if you thought to yourself, coniferous versus deciduous, you would be correct, right? Aside from the different latitudes, we have to think about the species composition, right? There's going to be a different mycorrhizal fungi community in the coniferous forest versus the deciduous forest. Although they might share a couple different species, largely they're going to be more specialized and specific to the types of trees that they're partnering with. In addition to that, we want to think about the microclimates that might exist around Lake Superior. There will be a map here in a second talking about that. But then in addition to that, we also have the different northern latitudes that my sites were located along. So we just want to make sure that we're capturing the different local adaptations that trees and mycorrhizal fungi communities might have in combination with one another. This is a smaller version of that diagram earlier, but we have the rhizosphere with baby white pines, and then the mycorrhizosphere. And it's actually quite interesting to see the difference in size of the root systems, where the ones without colonizations, those root systems will get long and gangly looking for their nutrients because the soil is sterilized and I don't fertilize to encourage the mycorrhizal associations. Whereas these root systems will often stay smaller, but the mycorrhizal partners will be reaching out and getting any sort of nutrients they can. This is an example of what an uncolonized root tip looks like. So you can see all the little root hairs coming off of the little root tips. And you can also see how short the root tips are. Versus mycorrhizal root tip colonizations. These are two different species. I don't know which one because I didn't sequence them, but these are just these are pretty typical morphologies that you would see, I would see under the microscope. But you can see these coralloid structures that kind of deform. So the root tips are inside of these little coralloid structures, but it encourages this kind of crazy growth. And then of course the mycelium, which is much finer than a root hair coming off of that colonization. And as stated before, they aid in nutrient uptake, drought resilience, and that soil stabilization, which is really great, especially in a changing climate. So this is what my experiment was conducted in. This is the Minnesota Lake Superior coastal zone. And within this coastal zone, the cool air of Lake Superior has a large effect on these forests. So oftentimes people collecting seeds for the state nursery or other nurseries, whether private or public, they will not collect within that zone because those trees tend not to do well in the interior of Minnesota because they're used to that cooler climate along Lake Superior. Nevertheless, we need to make sure those forests are succeeding and keeping up with kind of the climate change, or at least keeping a pulse on what they're doing in accordance to climate change. So my hypothesis is that seedlings inoculated with home soil would perform better than seedlings inoculated with away soil. And what does that mean? I will tell you. So we have 5 different sites. Duluth is quite long, so these 2 are considered Duluth, even though they're at opposite ends of Duluth. Castle Danger, Tetagooch State Park, Sawbill, which is the Sawbill Trail, and Judge Magny State Park. I took seed sources and soil sources from each of those 5 different sites. And then I also had control, which was just plain sterilized soil. I did a common garden experiment in the UMD greenhouse. And I took soil from one of those sites and applied it to all the different seed sources. And then I repeated that for all of the different soil slurries and seed sources. This gave me 30 total combinations. It was quite a large experiment. I had the original experimental design ended up with 1,250 seedlings per species. So it's quite robust, and it took a long time to assess all of those roots under the microscope. What I found is that the above ground growth rate did not differ between sites or between soil sources. So what we're seeing here is each panel is a seed source, so that's a tree population. And then on the x-axis here, we have the inoculation source, and then each dot individually here, I have a bunch of dots hidden, don't worry. Each point individually here is the home inoculation source. So if my hypothesis were correct, we would expect that these points would have a estimated marginal mean growth rate larger than the rest of the soil sources. But we can see that that's not necessarily the case. In fact, sometimes even the lowest growth rate, depending on which population it was. And even sometimes the controls, like in Castle Danger and Tetagooch, will have a higher growth rate than the rest, which is quite puzzling. But when we look at below ground, there's a slightly different story. So looking at percent root tip colonization only, I did do root to shoot ratio, but I won't be discussing that here today. When we look at estimated marginal mean percent colonization for just the soil slurry, the inoculation source, as a main effect, we can see that Judge Magny had a higher percent root tip colonization than the other sites. And then when we look at percent root tip colonization for the tree population, both Tetagooch and Sawbill had a higher percent root tip colonization than the other sites. But when we look at them as an interaction, there are no significant effects, even though we can kind of see that Judge Magny State Park overall had a higher percent root tip colonization. It wasn't enough to be significant. These are just things to kind of consider, but it doesn't necessarily give me what I was hoping to find, but that's science, right? Overall, in conclusion, and these are different coralloid structures, which is super cool. This is Stenococcum geophyllum, which is, it always looks like that, whether it's on white pine or yellow birch. That's an aside. Basically, select tree populations with desirable traits. That seems like a no-brainer. And select inoculum sources with diverse fungal communities compatible with the target tree species. So basically, be choosy and choose healthy forests with healthy trees and healthy fungi. Seems simple enough. But now we're going to take this and apply it to a more large-scale experiment and see what happens there. So what happens with mycorrhizal fungi after disturbances kind of depends on who or what the disturbance was. So it can help forest recover or get a jump start in reestablishing seedlings after disturbances if there are trees left in place. So if there's remnant trees retained from forestry practices or newly planted trees that were harvested and transplanted from a healthy forest, this might be able to help. But if that's just clear cut and there's nothing left to help reestablish, or there aren't any reference forests nearby to help distribute spores back into that area, that might be a problem. And the mycorrhizal fungi will search and move throughout the soil for only so long until it gives up and dies. There's also a post-wildfire where mycorrhizal fungi that specializes in wildfire areas might be able to dominate. And if you inoculate trees with mycorrhizal fungi that specialize in the post-wildfire environments, that might be more favorable. They help stabilize soils by producing those aggregates, those enzymes that bind soils. And they also break down nitrogen in the soils that might be in excess after a wildfire. So basically, in my opinion, commercial mycorrhizal fungi, as far as trying to implement it in a forestry scenario, is a no-go due to all of the risks that come along with it, as well as the high cost. Harvesting soil is quite costly and destructive, and there are many assumptions made. So you can't go around harvesting a bunch of soil from healthy forests because that will obviously quickly degrade the forest. And then you're also assuming that A, mycorrhizal fungi is there in the first place, and B, that there are no pathogens accompanying that soil transfer. And then transplanting trees from a non-disturbed area is both costly and time-consuming. You have to have people come out there, dig up the trees. You have to make sure the trees have adequate transport and everything they need to make sure that they don't die. And sure, on a small scale, it might be possible, but when we're talking about large-scale habitat restoration, this isn't necessarily feasible. So there are potential solutions. We have different types of dispersal. Tree to tree is where you might have a little tree neighboring with mycorrhizal fungi, neighboring a tree that doesn't have any mycorrhizal fungi. And then that fungi will kind of naturally spread to that other tree as mycelium does move throughout the soil and then colonize that tree. Then there's also alternative dispersal methods. So there's small mammals who eat the fungi and then poop the spores out. There's large mammals, predators, that eat the small mammals that then disperse the spores later on. There's range, so if fungi is, if it gets swept up in a rainstorm and then rains down, that's another dispersal method. And then of course we have wind. But for my project, we don't necessarily know which is doing what, so I'm calling it alternative dispersal. For tree to tree, I'm trying to figure out if we can use inoculated trees alongside uninoculated trees to help facilitate that. How many trees, inoculated trees to uninoculated trees do we have to have in order to have beneficial effects in the short term? So basically, how many do we have to help the seedlings establish themselves? I'll be using quantitative DNA to determine if the transfer was successful. So harvest these trees and see whether or not the fungi from these trees ended up right here. And I'm also looking at physiological traits to measure the effects to see if there's actually any benefit to the seedlings that were uninoculated and then presumably became inoculated. This is using a live inoculum technique, which this is versus like the propagules that are used in commercial mycorrhizal fungi mixes that are just dried products. This is similar to the buckthorn plug inoculation that Ryan was talking about earlier. But it's similar in the sense that it's live and it's active. And then there's alternative inoculation. So what's already in the soil? What was already there before I started planting my trees and inoculating them? What's coming in through small mammals, wind or rain? I'll be using quantitative DNA again for those soil samples. And then again, physiological trace to measure the effects. This is a diagram of what the experiment looks like. So X is nurse or colonized trees. These are ones that I pre-inoculated prior to going out into the field. And then the big BAM sign are the bait trees. Those were not inoculated. I purchased them from a nursery. And basically, I'm just trying to see one-to-one what's the effect of mycorrhizal fungi moving from this tree to this tree. 1 to 3. 3 was just an arbitrary number that I chose just to figure out, okay, what's this ratio? Or is there a difference between the effects of mycorrhizal fungi moving from this tree to these 3 trees? I have bait only to try and see what's coming in from the outside, what's inoculating the trees without any of my influence. And then nurse only just to kind of have a control. Last summer, I grew these trees in the pots, and then I overwintered them in a root cellar. And now I pulled them back out, and they'll be growing for a full another season. So they have 2 seasons under their belt prior to harvesting them this coming fall. This is kind of what the experimental design looks like in real life. So you can kind of see here the 1-to-1, 1-to-3, singular, singular. And there we go. So you can tell these trees are quite a bit further along in development, which might actually help them in regards to accepting that mycorrhizal fungi and benefiting from them. And then this is kind of what they look like on their pallets. I literally looked at all the trees that I planted that were nurse trees to determine whether or not they were colonized, and these are indeed little colonizations. These are forkings, and I call them crusts, but probably Pezizaceae family, so the cup fungi. And then these little forkings are kind of small and golden. This is what the plot looks like. So I have that nice hardware cloth to keep out the rodents. They still get in there and dig stuff up, but it discourages them. I have all of the pots on pallets because I wanted to keep the ground fungi from traveling up into the pots themselves. And I have a couple of pots off to the side that actually did happen. So that was a nice, like, hey, way to think about the future. And I have 12 pots per pallet. Soil sterilization. This is a big problem, right? So obviously I wanted to give myself the best chance of not having any outside fungal inoculum. That is indeed a turkey boiler, and that is a flamethrower. This was to try and flame sterilize the soil, but not overcook it. And then these are front end loaders. This was quite the day. And that's a really big pile of soil. But it didn't work. This is how much soil we got sterilized after like 2.5 hours of trying to sterilize. I'm like, wow, that's just not going to work. So on to plan 2, plan B. We ended up just doing kind of the classic, spread it out on a big black tarp and let it cook for several weeks. Kind of funny. Looking back at it, it was quite frustrating in the moment. But then I had this infrared thermometer to try and see what the soil temperature was, as well as thermistors within the soil to monitor what the soil temperatures were. And it didn't get up to what I had hoped, but it was better than nothing. All right, so what could this tell us? This could tell us whether or not it's beneficial to raise mycorrhizal fungi or raise inoculated seedlings with mycorrhizal fungi and then planting them out along trees that are being planted for reforestation efforts. So not necessarily blanket inoculating, but commodity item inoculating, trying to figure out whether or not it's worth it. And if it's not, it's not, but at least we know. And are the seedlings recruiting what they need to via alternative dispersal? Are these seedlings, the bait seedlings, are they getting what they need without any outside influence? And is it dependent on the surrounding environment? So the plot that I had was kind of surrounded by trees. It's shaded to try and have ideal growing conditions. And there's also fungi that is nearby. In a clear cut, would this be the same story? We don't know. So this is kind of where my research is at right now. As far as acknowledgements go, first of all, I'd like to thank the Minnesota Mycological Society for helping me fund this research. It'll be really helpful when it comes to the DNA assessments this fall. My committee, Dr. Julie Ederson is my advisor, and then Michael Joyce, Amanda Gruce, and Shanlan Chun have been extremely helpful in this process and experimental design. All the DNA help that I've received, all the seed and data collectors, and my army of undergraduate students that haven't been just mine. We've shared them in our lab, but it's been quite the effort. Measurement days and microscopy work has been pretty insane. So yeah, thank you for listening. Any questions?
Ryan has a question in the chat, and he's asking about, and Ryan, feel free to unmute if you'd like, but asking what species of fungi you inoculated the nurse trees with.
Yeah, so I inoculated with 2 different species of Swillus fungi that I collected and cultured out. So they were mycelial inoculation, not spore inoculation. However, they were in a grow chamber and they got cooked. So I didn't have the chance to test them prior to going out into the field. So I don't know if it was the Pizziza that went out into the field or if it was the Suillus that went out into the field. I do have a offshoot. I do have an offshoot experiment going on in the greenhouse right now. That is the second part of this. I had 30 leftover bait trees, and I still had my mycelium cultures in the grow chamber. So I inoculated those, and I'm about to inoculate more seedlings with that to see what those are like. But yeah, until I harvest all of the seedlings this fall, I won't know exactly what went out there and what I'm getting. If that makes sense.
Yeah, cool. I know it's tough to get pure cultures of ectomycorrhizal fungi. So congrats on getting those Swillus to go.
I don't know if they're pure either because I just sequenced them from the greenhouse thing, and like 3 species of Swillus came back, and I've been like keeping these cultures for 3 years. And they don't look contaminated, but I mean, pretty crazy. That's a different question.
Did it seem like when you saw them growing, could you tell, or could you kind of be like, oh, is that a no-no to try to guess ahead of time that like, gosh, these seem like they're doing better than that one? Because when I was looking at the pictures of all your plants and pots, it's like they did seem like they were doing better, the ones that were with the mycorrhizal bait or the host.
Yeah, so let's see if I can. That's not a great. There we go.
That one, yeah.
Yeah, so I haven't been able to analyze the data at all because some of them have great growth and some of them don't necessarily. But it's not a stark enough contrast to just be able to tell by eye. There's also there's maternal effects, right? I have no idea where these trees came from. Like, were they from the same mother or not? And for me, for my big cross inoculation experiment, there were very clear differences that were significantly different between populations. Like, some of the populations were just taller than others. But when I kind of normalized that, when and looked at just growth rate, it wasn't significant. So yeah, it's kind of important to know these things, and unfortunately I don't necessarily know that. So I'm hoping my sample size is big enough to help me decipher that, like what's real and what's not.
Awesome. Yeah, I have a lot of very, very mature oak trees in my yard. So of course they plant, or the squirrels plant, all sorts of little trees, and I feel so bad pulling them. But I've never really had good luck if I've tried to transplant them. It's earlier in your presentation when you were saying how hard it would be to transplant trees. They don't seem to do that well once you've disturbed them when they're small, unless they're
Yeah,
really small.
yeah, it really depends. Yeah, it just depends. Like, sometimes some things respond well and others just don't. Or they could be doing well for a few years and they just quit.
Mhm.
Um, but yeah.
Awesome. Do other people have questions? I think that's super interesting that you analyzed the commercial myco application, right? It's kind of like, what did they decide to put in there, and what's the combo? It's almost like they could have things that were competing with each other,
Yeah, yep.
right?
There's a bunch of different inocula out there. Unfortunately, I don't have the time or funding to explore all of those, but
Mm.
I think, you know, moving towards trying to create native focused inoculum, or just regional, or and helping people, if I can, find their own native combination of mycorrhizal fungi, would be really, really satisfying.
That's cool. Yeah, Peter, do you want to unmute?
Yeah, I was wondering if have you seen any research talking about tree to tree propagation from in actual forestry practices that seem to have effect? Leaving host trees or nurse trees uh, in clear cuts and things like that that have shown impact.
Yeah, so let me go back to this slide. There we go. Okay, so this Policelli et al. 2020 paper really touches on these two figures. Actually, it's part of the same figure. But they did a meta-analysis of a bunch of different studies that look into that, and it's absolutely worth looking at. It's pretty inspiring. But basically, if you don't leave those little islands, kind of what they summarized, if you don't leave those islands, there's not going to be any reestablishment unless there's dormant spores in the soil, or if stuff gets moved into that area. And so it's kind of dependent on how tolerant the mycelium is to that wildfire burning. How deep is it? How deep was the fire? With clear cutting, are there any kind of reservoir trees or reservoir, like little islands of trees left over to help those species? Because the mycelium will travel throughout the soil, but just not super far before it expends all its resources and dies. So
Yeah.
does that kind of answer your question?
Yeah, that and have you heard of anybody trying to encourage those techniques in the Midwest?
There are forestry, so not necessarily from a mycelium perspective, but from a habitat for like nesting birds perspective, and I mean also like grazing on ungulates like moose and whatnot. Basically anything but clear cut is best, but it takes a long time for those industrial practices to adopt new methods, even if the science is showing it. So basically, all we can do is show that it's worthwhile and somehow show that it helps forests recover better, therefore it helps your money recover better, you know. Sorry.
Well, you know, and like you point out, there's other benefits to those kind of things, not just, you know, forest health benefits directly or indirectly through, you know, fungal relationships, but habitat relationships and diversity and all kinds of other stuff that, you know, clear cut is kind of a little bit, I'll call it scorched earth without the fire.
Yeah, it's pretty outdated, and it just, I mean, nobody can walk into a clear cut and say this is great. Money is what makes the rules, yeah. Unfortunately, yeah. But that being said, you know, hopefully mycorrhizal fungi can help illuminate better forestry practices that then, you know, maybe there's more productivity there, you know, something like that to kind of motivate that direction. We'll see.
Well, thank you so much. Thank you both.
Thank you.